Hello, everybody. It's Noah for GCSE. And if you can hear me, just let me know. That will be great. It's so lovely to see you guys all here. Hopefully. And yeah, I'm so excited to get this live stream started. So I know you guys have had your RS exam or your RE exam yesterday. I hope that went, or today, sorry. I hope that went really, really well. A lot of you are saying that some of the questions actually came up in the live stream, which is amazing. Okay. So hopefully, I presume a lot of you who are at this live stream have watched the previous biology live stream that we did, which is on the left-hand side on the Tuesday, 7th of May, where we've covered those topics on the left-hand side. And that is because on the right-hand side, we're going to be going over the second half of the topics. So hopefully you have seen those, seen that live stream. If not, you can maybe watch it at the end of this live stream. Thank you guys for telling me that I'm not muted. Yes, so hopefully you can see the screen. Yeah, perfect. So what we're going to do is we're going to go through these questions on the right-hand side, and then at the end of the live stream, because that will probably take us an hour, so that probably take us till 7 p.m. And then at 7 p.m., this is for combined, higher, and triple, by the way. Okay, and then at 7 p.m. we're going to look at some past papers and go through them, and also look at, it'll be more of a question session where if you have any questions on topics in biology you don't quite understand, we'll go through those. Okay, let's now go through these questions. Thank you guys for attending, and you've left such nice comments, which makes me feel so happy. So thank you for that. Let's begin by answering the question. So the first topic is leaf structure. So how this is going to work is I'm going to read out the question. You guys can put some answers in the chat, and then I'll uncover the answers so you can make note of it if you want. I said combined, higher, and triple. So you can use it for foundation, but it's just it's going to be harder topics than you need. So I don't want to divert your attention away from what you should focus on. That's why I'm saying it's better for combined, higher, and triple. All of this content is required for triple. Combined, higher, a little bit less, etc. Okay, so let's get started with this. The changes in the mean width of the stomata in normal conditions are an advantage to this plant. Explain how. Okay, so what you're talking about is the stomata sizes, and you're being asked, why is the stomata being there an advantage to the plant? One second, I'm just getting the mark scheme ready. So firstly, you need to talk about the structure of the stomata and how that works. So the stomata closes at night because there's less or no photosynthesis, there's no light. And at the same time, stomata opens during the day. So as stomata closes during the night, it prevents any excess water loss for which there's no photosynthesis happening. Okay, they're using words like mean width to try and confuse you initially, but don't get too caught up on that. Just think the purpose of stomata opening and closing. Okay, so stomata opens wide during the day in order to maximize the photosynthesis, because obviously there's daylight and there's sunlight, so they want to get as much water across for photosynthesis. Perfect. And I see a lot of you are saying also because stomata opening wide takes in more carbon dioxide for photosynthesis. And yes, that's another point. So here's the mark scheme if you want to take a note of that. Okay, let's now move on to the next topic, which is transpiration and translocation. By the way, these questions aren't just randomly picked. These are the questions that most students got incorrect or make mistakes on. So that's where these are coming from. Okay, moving on to the next one. Transpiration and translocation. So the first question, define and explain the process of transpiration. Okay, so this is talking allowing nutrients wouldn't get the mark because you're talking about water, because stomata only lets out water, and sometimes it lets a bit of carbon dioxide in as well, but it's mainly for water. Okay, so for this next question, explaining the process of transpiration. I'm glad it's helpful, guys. Thank you. So the movement of, you need to talk about the transpiration stream. Now, the transpiration stream is literally the movement of water from the roots to the shoots, taking water, it's literally being drawn out of a plant. If you can imagine a pipette is basically sucking out the water of a plant. That's the same thing. Now, it's not just water, it's also mineral ions. Okay, it's really important you say from the roots to the leaves or the shoots. And how is this happening? It's happening via the xylem tissue. Now, I can see people are saying root hair cells. Yes, that's what starts it off. But so the root hair cells are in the root, and that then allows water to enter via osmosis. Okay, and then water then goes up the transpiration stream. But it's really important you talk about how it goes up the transpiration stream. Okay, it's because of a transpirational pull. As water is being evaporated in the leaf from inside the cells, this causes it to almost be sucked up like a vacuum. Okay, and you want to talk about the xylem cells, and you might want to talk about some advantages and adaptations for xylem cells as well. That would be super helpful. So there's the mark scheme for that question. Moving on to the next one. Well done for all of you guys who are putting your answers in the chat as well. I think that's a really good exercise, and a lot of you are learning from your mistakes and getting a lot right as well. Okay, compare the process of transpiration to translocation. So we just looked at the process of transpiration. So in transpiration, you've got the xylem tissue transporting water from the roots to the leaves and the stem. Now, transpiration is a one-directional flow. It's going from the roots to the shoots. The movement of water caused by the evaporation leads to the transpirational pull, drawing out more and more water from the roots. Whereas in translocation, it's multi-directional, okay? So it's happening in different directions. And another thing you can talk about is transpiration is for water and mineral ions. Translocation is for dissolved sugars. Well done for the chat. I'm reading all of your answers and they're all correct. Okay. So you can also talk about the different types of structures of xylem and phloem, as I saw you guys saying in the beginning. So phloem are not dead, whereas xylem cells are dead. Xylem cells have lignin, which are spiraled into their cell walls. Yes, Jude, that's right. I'm glad to see you're here. Yep, they're joined end to end. Okay. And you can also say that both are essential for plants to grow and survive. Okay, because you're not just talking about the differences, you can also mention the similarities as well. Next one. Describe 3 reasons why transpiration is important. Okay, so firstly, we need to think about the purposes of transpiration. It transports dissolved mineral ions. What are dissolved mineral ions important for? Well, they're key for plant growth, looking at magnesium, looking at nitrate ions, etc. They're used to make things like chlorophyll, which is integral for photosynthesis. This will be available immediately after as well, by the way. Okay, now water is required for photosynthesis, and therefore transpiration that's another reason why it's important. And another reason why transpiration is important is because the evaporation of water actually cools the plant down. Perfect. Hopefully you guys have got that. And if I am going too fast, then do let me know. If not, we'll keep going at this pace. So let's now move on to the next topic, which is very different. It is bacteria culture, the required practical. Okay, a lot of people forget this required practical, so it's really important to go through this. So with the required practical of preparing a bacteria culture, too fast. Okay, I'll go a bit slower. And what I'll do is, I'll upload these slides definitely to my website. Okay, thank you guys for telling me. So, the bacteria culture. So the bacteria culture, you need to split the practical into the different parts. So you've got the pre-inoculation, the inoculation, and the post-inoculation. So talking about the pre-inoculation, this is getting everything ready for the bacteria required practical. So a lot of sterilization has to happen because schools won't just let you grow a bacteria culture. Okay? Thank you for the compliments. And schools wouldn't just let you grow a bacteria culture because you could end up with some really harmful things happening. So you need to ensure that there are some safety preventions for any risks that could happen. And talking about sterilizing things. So firstly, sterilizing the petri dish or whatever thing that you're using. Sterilizing the gel. And what you're sterilizing it for is to kill unwanted bacteria. Another thing you want to do is you want to pass the inoculating loop, which is effectively that piece of material that you use to actually transfer the bacteria. So you want to pass that through a flame in order to sterilize it, in order to ensure that there's no other bacteria lying on that inoculating loop. Okay, so that's all of the sterilizing points you have to talk about. This is for triple and combined. Higher. Okay, now looking at the inoculation parts of things. So you're going to use the inoculation loop to spread bacteria onto the agar gel of the inside the petri dish. You're then going to close the lid of the petri dish to prevent any microbes entering. But you're also going to close it to prevent the bacteria escaping. So it's twofold. Okay, now looking at post-inoculation. So post-inoculation, once you've done that, you want to seal the lid onto the petri dish with tape to prevent any microbes entering, and also you're going to place it upside down. You want to place it upside down so that any moisture that might fall onto the bacteria, instead you're flipping it so that no moisture will fall onto it, and therefore you're having controlled conditions. Okay. And you're also going to have it at 25 degrees Celsius. And this is the maximum temperature. Now, you don't want it too high and you don't want it too low. The reason why is because if it's too high, you could actually cause the bacteria to die, or you could cause bacteria which is actually quite harmful to grow. And if you, so that's it. That's all you have to put for the bacteria required practical. I hope I'm not going too slow now. I know a lot of people are saying to hurry up, so it's a bit difficult. I hope I'm going at a good pace for most people. Let's now move on to the next one. Viral diseases. Okay, explain why TMV causes stunted growth. So tobacco mosaic virus, why does that cause stunted growth? I'm not too sure which topics are not in combined. This is not only combined, this is for triple, but it's good questions for combined higher. But if it's not part of your specification, then just don't worry about it. Okay, so explain why TMV causes stunted growth. Well, one of the things that happens is the leaves become damaged. And because the leaves become damaged, and the chlorophyll is becoming damaged, that means that the plants can't photosynthesize anymore. If they can't photosynthesize effectively, they can't create food. They can't create food, then they can't create then they don't have energy. So anything that requires energy, and via physics, you know that all work requires energy, the plant can't grow, etc. Another thing you can talk about is the plants therefore don't produce enough sugar for optimum growth. So that's what we were saying. Since this is only a 2 marker, you can talk about those 2 bits there because we're focusing on stunted growth. We are going to go over every topic in the second half. If you wanted to do the first half, then you want to go over the first live stream that we did on Tuesday. Okay, moving on to the next one. Explain how measles spreads from one person to another. Okay. Now, a lot of students got this wrong when it was actually put in a paper, so let's see if you guys know this one. So, measles, how are they spread firstly? Measles are spread via the inhalation of droplets in the air. That is how measles are spread. You're expected to know how these different things spread, whether it is a sexually transmitted disease or it's something like this. And then, so that's your first point, that it spreads via the inhalation of droplets of measles in the air. Your second point is then from sneezes or coughs of the infected person. So it kind of splits a sentence up into 2 marks there. Okay, we're now moving on to the next topic, which is bacterial diseases. So we've got a section on each of the different types of diseases. That was a viral one. Let's now move on to bacterial diseases. So TB, so this was a context question, so you were giving a bit of backstory before you were supposed to answer this one. So TB is a bacterial infection. Explain why people living in a densely populated area are more likely to be infected by TB. So since it's a bacterial infection and you're told that you're more likely to be affected if you live in a densely populated area, that suggests that it might be spread via the inhalation of droplets, just how we spoke about previously. And therefore, if there's a greater population, there's a greater chance of droplets which contain TB in the air, causing you to have a greater chance of inhaling it. Perfect. Now, moving on to the second part of this, give reasons why a person infected by a small number of TB may not actually develop the disease. So what you want to say here is that the white blood cells produce enough antibodies really rapidly to destroy the pathogen. So before the pathogen can reproduce sufficiently to cause any symptoms, your body starts producing enough antibodies. So you end up not even feeling the symptoms. And that's what you call a strong immune system. So I hope that explains those topics. Okay, and let's now move on to the next slide, which is fungal diseases. Describe how plants become infected with rose black spot disease. Okay, so this is just a 2 marker, so you're expected 2 kind of sentences, and you start off by saying rose black spot is a fungal disease. Okay, and that is literally 1 mark there. And then the second mark is its spores are spread via the wind or in water droplets. And that's what you're expected to know, how it is transmitted. Now, all fungal diseases are spread via spores, so that's really important. And yes, they travel through the wind. They also travel through water as well. So well done for everyone saying that. Okay, moving on to the next one, which is protist diseases. Evaluate the different methods to reduce the spread of malaria. Now, people often confuse malaria as a virus, but in fact, it is a protist disease. Okay, so there's a lot of points that you could make here. So you could talk firstly about non-insecticide treated nets and insecticide treated nets. So what that means is often people who live in countries where malaria is rife, what they do is they actually sleep in nets, and these nets can have they obviously act as a physical barrier so that these protists, these vectors, cannot come to you and obviously transmit malaria. And a way of strengthening that is by spraying insecticide onto those nets, because that then kills the mosquitoes as well. Another way you can talk about, I can see people put lots of valid methods in the chat. You can talk about getting rid of standing water. So what happens is mosquitoes breed really heavily and rapidly in water body sites. So what they do is they get rid of these areas of water that are close to where a lot of locals live. And they actually did this in Brazil, and it ended up working. Another thing you can say, which I've seen as well by Jeremiah, wearing long sleeve clothes, that is a really good way of doing it as well. Obviously, sometimes that doesn't work all the time, but you don't have to talk about how effective it is necessarily. Another one is closing windows and doors. Another thing you can say is taking anti-malaria drugs up to 4 weeks before you visit a country which contains malaria. Sometimes that is required to do. I see people asking, would a question like this actually be 6 marks? Well, in fact, it was a question that was 6 marks. So these are the actual real questions from the exam. What I've done is just tweaked it by adding a few more possible answers so that you guys are well equipped for the exam. Okay. And having more malaria tests is another way of reducing the spread of malaria. Okay, so I hope we went through that question well, and let's now go on to the next one, which is talking about the human defense system. Vaccination, yep, that's what I talk about with the anti-malaria drugs. But again, vaccination towards malaria isn't always effective, so it depends. Okay, so looking at the human defense system, question 1. Describe some non-specific defense systems. Now, a lot of students panicked when they saw this, when they saw the word non-specific, but that just means that it's everything that's not white blood cells, that's not your internal antibodies being produced, it's everything external. Okay? So, for example, your nose has cilia, which are little hair projections. These cilia trap pathogens. And also, you have goblet cells, which make mucus that ends up trapping more pathogens. So it's actually quite difficult for pathogens to be inhaled via your nose. And that's why you don't really hear about people catching diseases via their nose. Okay. The skin. The skin acts as a physical barrier. There's also some antimicrobial secretions. So you've got cells in your skin which release kind of this oil, and you probably have realized that. And that's called sebum. And that is actually antibacterial. So that is a non-specific defense system. Another thing you can talk about is the trachea and bronchi. So the trachea and bronchi also, just like the nose, have cilia, and they also secrete mucus. And the cilia doesn't just trap the pathogens, but they also move so that they're wafting the pathogens to be swallowed by the stomach and the hydrochloric acid that I saw people have mentioned as well. Now, Jed, when you said white blood cells clot the blood, it's actually the platelets that clot the blood, and both of those are talking about specific defense systems. Okay. Now, continuing with non-specific, you've got the stomach, as we spoke about, with the hydrochloric acid that kills pathogens, and also eyes. So tears actually have enzymes to kill bacteria. So tears have antiseptic properties. Okay, so that's why doctors sometimes recommend that it's actually good to cry. It's acting as an extra barrier. Yes, this is my real voice. It's not, I don't know how it wouldn't be, but yeah. Interested to know why you're asking that. Okay, so that is the mark scheme for that question. Moving on to the next one. What are the 3 ways white blood cells protect us? So, the first way they protect us, producing antibodies. So once these antibodies bind to the Cheyenne, that's your shout out. So the white blood cells produce antibodies, and once the antibodies are produced, they are a complementary shape to the pathogens. So it's just how enzymes work with the lock and key theory. So they bind to the pathogens, they clump together, and white blood cells then find them easily. Okay, moving on to the second part. So the second part is that white blood cells produce antitoxins, which do the similar thing to antibodies, but in fact they pair up to toxins. That toxins are the things that when we have a bacteria inside of us or any sort of living organism that's trying to intend us harm, toxins are the things that actually make us ill or make us feel sick. Okay? And the last one, which is phagocytosis. So what happens is the engulfing of the cell, engulfing of the bacteria or the bad cells. Now, another thing we can talk about, which isn't actually part of the 3 ways white blood cells protect us, but more about the specific defense system inside of us, and it's spoken a lot in the mark schemes, is memory cells. So memory cells form, and they then have a rapid response if you ever are to encounter the pathogen again. So if the pathogen ever comes back into your body, that's why you are supposed to be immune. But obviously with COVID, the difficulty was there was a lot of different variants and mutations. So you could have COVID multiple times because your memory cells wouldn't exactly work every time. So that is the mark scheme for that. Moving on to the next one. Explain how platelets work on a wound. So this is what we were speaking about. So, explain how platelets work on a wound. What happens is you get a cut somewhere or you get injured somewhere and there's the exposure of blood. So what happens is I'm giving you a lot of detail for this just to help you understand it and visualize it, but you only need 4 of these points I'm going to speak about, okay? So, you've got small blood cells, your small blood vessels, your capillaries. What happens is if you're cut or injured somewhere, some of the vessels are cut and they then allow blood to leak out. Okay? Now, the tissue surrounding that cut then begins to swell. So I don't notice if you've ever cut yourself, your skin begins to swell slightly in that area. Okay, now as it swells, what it's doing is it's trying to narrow the capillaries to stop excess blood leaking. The next step is platelets. So platelets are all around your body because they can't obviously anticipate where you're going to get hurt. And then what happens is once that area starts to get inflamed, your skin, all of the platelets run to that area. And what they do is they then form a net-like structure. They stick to the edges of a wound and they then work towards the center, therefore plugging a wound. And that then forms what we call a scab. Okay? And I've got a term here, soluble fibrinogen, which you don't need to necessarily know, but that's just a key word. And that is the strands of fibers which tangle around the plug to make it stronger. So the scab is often referred to as a plug in biology. Then what happens, therefore, is, well, why is it plugged in the first place? Well, there's multiple reasons why we want to create a barrier. Firstly, we don't want too much blood going out because then that means that we are losing blood and that could threaten our life. Another thing is that you don't want any bacteria or pathogens entering your system, and therefore that plug is really important. Now, the plug eventually dries out to form a scab, which provides protection, and they often say don't pick your scabs because new skin is still being formed, so it's really important for that to happen. Yes, platelets are important for clotting as well. So that's what we were speaking about. When they form that net-like structure, that is called clotting. So blood clotting is important. But if your blood starts to clot when you don't have an injury, that is not good because it can block oxygen from getting through via the red blood cells. Okay, so I hope that is helpful, and we're now going to move on to the next topic, which is vaccination. I hope I'm going at a good pace for you guys to understand now. 1 second. Okay, now moving on to the next one, which is describe the benefits to the individual, the family, and the wider community of a parent's decision to vaccinate their child, and why some parents wouldn't. So, reasons why people would vaccinate. The individual would want to vaccinate because it prevents the spread of any diseases, and they may feel selfless and they don't want to continue to spread it, and there may be no cure for it. Now, this specific question was for measles, actually. So, the mark schemes might be a little bit towards measles. However, most children have already, so they also want herd immunity. That's another reason as well. Um, for the family, they may choose to do it because they can't take time off to look after the loved ones who have measles. They don't have the ability to do that, and therefore it's important for families to get vaccinated. Um, and they might not also want to see their family members suffering as well with the disease. That's another reason why they'll want to get people vaccinated. If you have a weak immune system, people in the household are not compromised as well. Herd immunity is the idea that if some people, or if the large majority of people vaccinate themselves, then that means that even the people who are not vaccinated, they are also protected. Yes, as Roro said, that's a really good explanation. Moving on to the next one, the wider community. So there's less chance of an epidemic as well, and the child won't be at risk to the pregnant or elderly or wider society. Okay, moving on to the next one. List some advantages and disadvantages of vaccination. So, vaccination advantages. Firstly, being vaccinated means it's actually eradicated many diseases and prevented lots of epidemics from happening. But some disadvantages include that it's not always been effective in providing immunity. People can often have bad reactions after taking it. Okay, so those are some advantages and disadvantages of vaccination. Also, yep, people have been saying in the chat as well, Nicole and Olive have been saying side effects. Yep, that's a really good one as well. The next one, explain why the vaccine causes immunity but not the disease. So we spoke about a similar answer to why you may not even feel the disease even though you've had it. Now, what you can say is the virus. So how can the vaccine cause immunity and not the disease? So what happens is you inject a very small portion of the virus that's weak or inactive into your body. Therefore, it does not cause the disease because the virus is simply too weak. It then stimulates your lymphocytes, your white blood cells, to produce a lot of antibodies, antibody production. It stimulates that. And then what happens is the memory cells remain so that the body can very quickly produce antibodies in response to any infection. So that is why the vaccine causes immunity, but you don't feel the disease because it's weak or inactive. Moving on to the next topic, which is antibodies and painkillers. So, explain the process of antibiotic resistance, how to prevent antibiotic resistance, and reasons it has increased over the years. So, this can be a 4 to 6 marker. So, antibiotic resistance is when people have been taking antibiotics, and what happens is the bacteria that are susceptible to those antibiotics are initially killed off. But some of the bacteria are not killed off, and you then stop taking the antibiotics because you stop feeling any sort of sickness. But in reality, those bacteria are still in your body. And what happens is those bacteria can then have formed resistance to it, and they can have undergo a mutation as they now know they've experienced contact with this antibiotic. They then know how to overcome it. They might create a mutation that therefore is immune to the antibiotic. It's a different shape. So the resistant bacteria then reproduce, and as it's survival of the fittest, they obviously reproduce more, and since they're surviving, their population becomes dominant. So what are some reasons why it's increased over the years? Because a lot of people have been not finishing the course of their antibiotics. So a way to prevent that is to finish the course of your antibiotics. Another reason is to stop over prescribing drugs, because that is increasing the possibility that antibiotic resistance could take place. Overuse as well, mutations of bacteria. Antibiotics only work on the non-resistant, so you want to prevent the chance of mutations of bacteria from occurring. Can I do an edX Cell Biology paper 1 live? I'm doing AQA. I don't have too much experience with edX Cell. I might create shorts for it, but I'm not sure if I can do an entire live stream. I'm so sorry. This one is for AQA. Okay, let's now move on to the next question. Okay, why in the past might people have chewed on the bark of a willow tree if they had a headache? So, what you want to talk about in these willow trees is the idea of aspirin. So this is actually a triple question, but you can still be here if you do combined higher. Okay, so aspirin is found naturally occurring in these trees, in these willow trees. And therefore it was used by the indigenous communities as a herbal remedy for painkillers. So I don't know if you knew this, but a large majority of the medicine we now use is because of these aboriginals who found out by trialing different effectively drugs from trees and sorts of plants like the foxglove plant. And it's because of them that we have so many cures to things. Now, penicillin came from an Alexander Fleming, and it was actually a mistake that happened. So what was happening was he was trying to, I forgot what he was trying to do, but what happened was it ended up that penicillin started forming, and he realized that this actually started killing lots of bacteria. There was no bacteria in that area, so he realized that this was a really effective drug, and therefore that's what happened afterwards. Okay, let's now move on to discovery and development of drugs. If you like history or if you do history, that might help you. Foxglove is a drug that is, you don't need to know about foxglove and digitalis, but maybe you've heard of them. And if you were to have a question on them, they would have to tell you what that's about. Okay. Moving on to the next one. So, why can't drugs for bacterial infections treat viruses? Okay, so think about that. Why can't drugs for bacterial infections treat viruses? So, what you want to say is that viruses are inside of cells. Okay, they hack inside of cells and they kind of terrorize them and therefore take control of it. So they are inaccessible to the drug, as it would then cause, if there was going to be a drug that was suitable for killing viruses, they would end up killing the cells, and that's not what you want to do. You want to protect them. Okay, and that is the answer. Moving on to the next one, explain the stages in a drug trial. So you start off with preclinical tests. These are on computer models and singular human cells. You then have the next stage, which is the preclinical tests on animals. Okay, so you've got the cells and the computer models, and then on animals. You then have clinical trials, which means trials on humans. So that's the difference between clinical and preclinical. Preclinical is before you do it on humans, and clinical is once you start doing trials on humans. So you then begin the clinical trials, but you start off with healthy volunteers. Okay, it's important you say that. You start off with healthy volunteers. They might actually ask you in the exam why they don't actually start the clinical trials on the people who have the actual um, virus. And the reason why is because the people who have the virus, we don't know what effects this drug could have. It could worsen their condition, further weakening their immune system and put them at risk. So you have to start it on healthy volunteers. Okay, so once you've done that, you've trialed it on healthy volunteers, you're then going to trial it with patients who actually have the relevant disease. And this is obviously assuming that it's past the previous stages. And yeah, a lot of people saying the same ages and gender, exactly. You want some control variables as well. And finally, after it's trialed with the patients, the drug is then distributed to a larger population. You don't have to mention double-blind trials because you're only speaking about the stages in a drug trial, but you can mention it as well. Moving on to the next one. Explain how clinical trials should be carried out. So the trials on humans. Now we can speak about the double-blind trials, etc. So starting off with, we know that it's initially given to healthy volunteers at a low dosage. So remember, it's important to talk about the dosage. So it's given at a low dosage. To test for toxicity, that's a really important idea, and to test that there are no side effects. The next stage is that it's then given to patients who have the relevant disease, as we spoke about. And you're then testing during that period for the optimum dosage, for efficacy, to check for side effects, to test if it works in a double-blind trial. Which means a double-blind trial is effectively when neither the patient nor the doctors know who actually have the antibodies and who is the placebo. Okay, we're now moving on to the next part. Give 3 reasons why we must trial drugs. Now, this should just be coming from your memory. You should know this like the back of your hand. So it's efficacy, dosage, efficiency, or toxicity. So you can speak about any of those points there. Moving on to the next one. What is the point of a placebo? Injecting a drug which actually has no effect on the individual. What is the point of it? Well, in the mark schemes that I've seen, they've always said the point of a placebo, is to act as a control. But I did see in one of the mark schemes that they asked this question, is it said to compare the effects of taking the treatment versus not taking the treatment. Now, even though that seems like the more logical answer, I implore you to use the answer to act as a control because it always, for some reason, is in the mark scheme. Okay? So, I hope you found that page helpful. We're now on to the next one, which is all about monoclonal antibodies. Okay. Evaluate the personal benefits of using monoclonal antibodies for diagnostic testing. Okay, so what are the possible benefits there? So, firstly, you want to say, well, what are the different uses for monoclonal antibodies? They're used in pregnancy tests. They are sometimes used in actually finding different STDs. So, let's talk about the advantages of that and the personal benefit. Well, we know that pregnancy tests, they are easy to use, they are readily available, and they are very accurate. Also with pregnancy tests and also testing for STDs, you might find it uncomfortable to go to a doctor and have yourself checked out and a GP. So there's no need to consult a GP. You can actually access it yourself. And another good point about that is therefore the GP's NHS, which we know is currently or constantly under pressure, it reduces extra things that people need to see them for. Thank you, Logan. I appreciate that. I want to see your guys' results because I know you're going to do really, really well. Okay, another thing is you can start to take care of your health of your fetus from day one because you know immediately after monoclonal antibody testing because you might put off going to the GP. And you can also and other advantages is that urine testing is quicker, it's less invasive, and you can therefore treat diseases or viruses from the beginning, from the onset. Okay, moving on to the next topic, which is explain how a pregnancy test works. So this is going into the specifics of that. So firstly, you want to say, as the urine passes through the pregnancy test, it goes initially through there are two zones in a pregnancy test, okay? You've got the reaction zone and you've got the control zone. So it initially goes through the control zone, and the control zone is important because it's showing that the pregnancy test works. Okay, so the control zone should always have one line or one red line pointing there. So if you're not pregnant, you're going to have at least one red line on there, or you're only going to have one red line on there. If you are pregnant, you're going to have two red lines because of the results zone. So for the results zone, what happens is the hormone that pregnant women have, which is called HCG, binds to the hormone in the results zone. So there's a specific the hormone antibody. So there's an antibody on the pregnancy test that will only bind to the pregnancy hormone. And then, therefore, if you are pregnant, it will bind to the hormone. And what happens is that those antibodies are often attached to ink and dyes. So then what happens is that dye then shows up. And therefore, it shows a positive result for pregnancy. Okay, moving on to the next one. List advantages and disadvantages for monoclonal antibodies. So advantages, it only binds to a specific cell. Okay, so monoclonal antibodies are very, very accurate. They will never bind to a cell that it's not got a complementary shape to, just because it physically can't. Another thing is that using monoclonal antibodies means that because it binds to only those specific cells, the healthy body cells won't be affected. So again, that's really good. Another thing is that monoclonal antibodies can be engineered to treat many different conditions. Now, disadvantages of using monoclonal antibodies. Monoclonal antibodies are often difficult to attach monoclonal antibodies to specific drugs. So obviously, not obviously, but monoclonal antibodies, they're trying to use them for treating cancer, and not treating cancer, but when you're undergoing radiotherapy, what will happen is the monoclonal antibodies, they attach them to the toxic drugs that are designed to kill the cancer cells, and they're only going to kill the cancer cells and no other cells. But it's sometimes difficult to attach the drugs to monoclonal antibodies. And another thing is, since monoclonal antibodies come from mice, it triggers immune responses because you've got a foreign cells in your body that can trigger immune responses. And on top of that, if you're allergic to mice, you can actually have an allergic reaction. And another thing is it takes time to create as well, which some people have said in the chat. Okay, now moving on to the last question of this page. Describe the steps of producing monoclonal antibodies. So how are they made? And this is where we're going to be talking about the mice. So what they do is they inject the virus into the mouse. So let's use an example, COVID-19. They inject COVID into the mouse. This then stimulates the mouse's lymphocytes, which is, remember, a specific type of white blood cells. It stimulates the lymphocytes to produce antibodies in order to fight against this virus. What then happens is the doctor then or the surgeon, whoever's doing it, the scientist, extracts these lymphocytes out, because we know that therefore they are successful in going and preventing the antigen from spreading, so they work. So you then take the lymphocytes out and you then combine the lymphocytes with a tumor cell. Now, what the tumor cell does is it then, we know about tumor cells, they undergo rapid mitosis at a really, really fast rate. So if we're doing rapid mitosis of the lymphocytes, that's a good thing. We're creating loads and loads of those white blood cells that work against the disease, or the virus, sorry. Another way you could do it is create a hybridoma. Now, you don't need to know how a hybridoma is made, you just need to know that's another option. Okay, and then finally, you're going to clone that cell further so that you can send it out to the population. Okay, so moving on to the next one. Plant diseases and defense responses. So, suggest how plants have evolved to defend themselves against pathogens and herbivores. No, this is not A-level biology, this is GCSE biology. Okay, so let's speak about this. So how have plants evolved to defend themselves against pathogens? There's loads of different ways this happens. So firstly, there's physical, mechanical, and chemical responses that can occur. So you can talk about the leaves. If they curl when they're touched, what happens is that often that deters herbivores, because if there's any movement, sudden movements from plants, it actually does make the animals or the herbivores very squeamish, and they then choose another thing. And on top of that, if the leaves curl when they are touched, that ensures that any insects that are taking a lot of the nutrients from the phloem in the plant, they then fall off. And therefore the plant is less likely to be damaged, and it's less likely to allow pathogen entry. Now, you can talk about specific plants. So the Acacia plants have hollow thorns, and a lot of plants have hollow thorns, which means that they have sharp thorns, but they're empty. And what happens is those thorns are actually colonized by ants. So if you do the Amazon GCSE geography case study, you might be aware of this. And it was actually asked about in the last year's exam about how the different plants adapt in geography. So, and then what happens is those ants live in the thorns of the plant. And you might think, how is that a good defense? Well, what happens is because the plants live in the thorns, if there are any, not the plants, because the insects live in the thorns, if there are any other insects that try and take food from the plant or try and hurt the plant, those insects that live in the thorns realize that they are ruining their home. And so they actually end up killing and getting rid of those. Now, another thing is the thorns themselves. They're sharp and they deter herbivores. They're also very large and obvious, and therefore it just makes it animals less likely to take any water from there. Also, hairy stems. So having lots of hair that have a bit of stinging chemicals in them, that makes herbivores think that the plant is rotting, and also humans as well, because we are a threat to them often, and therefore you don't really want to touch something like stinging nettles because that makes you uncomfortable. The bacteria culture is a triple practical. Yeah, we spoke about that earlier on in the live stream. Okay, explain. That is just the wrong page, I believe. Sorry about that. That the page moved, but this is now you can see the answers. That's fine. So, name some physical and chemical plant defense systems. So you can see there's cellulose in the cell wall, which just makes it, because it's very thick, it makes it a bigger barrier to entry, because a lot of the insects, they're using their sharp noses or mouths to try and inject themselves into there. And also, that's a good response because it prevents any, for example, mosquitoes, them spreading diseases to the plant as well, because that can happen a lot of the time. The outer dead cells on trees, those are the things that make bark, and that's a good protection for the trees. And infected leaves, they fall off to protect the healthy leaves, which is a really nice thing for them to do. Chemical barriers as well. So you've got the antibacterial chemicals that plants may produce. You've got the stinging chemicals. And yes, you've also got antibacterial chemicals as well to help actually the plant itself recover from anything that happens. I'm not going to do foundation. Okay, explain how a nettle is adapted for defense and protection. So the nettles have stinging hairs, which upon contact of the stinging hairs, they release chemicals. For example, something called formic acid. And this then leaves blurriness. This causes blurriness and rashes. And so if you've experienced that plant before, you don't want to retouch that plant. Another thing is the thorns that cause pain to deter the herbivore as well. Trichomes are interesting. So they're small hairs, tiny hair projections that are on plants, and they actually cause inflammation and appear very unusual and prickly. So those are the things that are on stinging nettles. Okay, we're moving on to the next topic now, which is photosynthesis. We're almost towards the end of these questions. So, name some uses of photosynthesis. So, photosynthesis, different uses. So, it releases energy in respiration, releases energy so that respiration can take place. Cellulose is therefore made from the products of photosynthesis. Also, products of photosynthesis are used for insoluble storage, known as starch in plants. Also to produce amino acids, as you said, make energy. But you want to talk about what things you can do with that energy as well. So respiration is a good example. And also to convert fats to oils as further storage, like lipase. Okay, describe how energy for the photosynthesis reaction is gained by plants. So what happens is, well, it's quite simple. It's just, this is really a 1 marker, not a 2 marker. And it is light energy is absorbed by chlorophyll. Vixie, that is a very good point. Well done for picking that up. So yeah, you can't make energy. You can only release energy. So it's a good point. Thank you for the correction. I'm so sorry, I don't know how to get moderators in the chat. I'm not too familiar with that. Otherwise, I would prevent spammers from annoying people. But I don't know how to do that, unfortunately. I will try and make the slides available for you guys. And as for a face reveal, that will be done when I get 100,000 subscribers. That's my aim for this year. So if you can help me reach that, then maybe. Okay, moving on to the photosynthesis required practical. Okay, so let's speak about the required practical. So what you want to do is you take a boiling tube, you then place 10 centimeters of sodium hydrogen carbonate, which is effectively this substance that produces carbon dioxide due to photosynthesis. Okay, and what happens is once you've added the 10 centimeters cubed of sodium hydrogen carbonate solution, what you're going to do is you're going to cut a piece of pondweed, which is a type of plant, and you're going to put the pondweed into the test tube. 1 second. Now, as you put the pondweed into the test tube, you're going to have the cut end, and you're going to put the cut end at the top so that that's producing the carbon dioxide bubbles. Now, what you're going to do is firstly, you're going to leave it for 5 minutes. You're going to leave it for 5 minutes as a control and leaving it to acclimatize. They might ask you, why do you put it in there? So why do you put the pondweed in there and leave it for 5 minutes before even doing anything? Because it's producing bubbles still. You want to ensure that it is allowed to acclimatize. The next stage is starting the stopwatch after 5 minutes. And then after 5 minutes, you're literally going to count the number of bubbles per minute. Now, if this is too much for you, what you can choose to do, and even mention this in your exam, is actually just film it and then you can record it after to make sure. And then what you're going to do is you're then going to calculate the mean across 5 different minutes. What you then do is you then repeat it with different distances of light. So what you can do is you can look at the effect of light on the required practical for photosynthesis. And you can talk about how the distance of a lamp to the pondweed in the test tube affects the amount of carbon dioxide released, the amount of photosynthesis that happens, and you can do it at varying lengths. Yeah. Okay. Suggest 2 reasons why the rate of water loss in both plants changed after 2.5 hours. You use sodium hydrogen carbonate because water doesn't produce any carbon dioxide, as people were saying. So the sodium hydrogen carbonate produces carbon dioxide, which allows photosynthesis to take place. Because in order for photosynthesis to take place, what 2 things do you need? Sodium hydrogen carbonate and water. Now the water can actually be made because you've got sodium hydrogen carbonate. They contain different substances in that, so you can have water in that substance and also the carbon dioxide. Moving on to the next question then. So suggest 2 reasons why the rate of water loss in both plants changed after 2.5 hours. So that was a specific contextual question. So what just happened was it was warmer. So you need to talk about the impact of temperature. Yeah, more humid, exactly. Or less humid, and the light intensity was higher, it was windier, etc. So you can talk about the different impacts. Did I just get £2 for no? Okay, thank you. I appreciate the £2. Okay. Moving on to the next one. Suggest one possible cause of the anomalous result. Well, you can say the value misread, or the lamp has been moved, or the pondweed hadn't yet acclimatized. So those are different reasons for possible errors occurring. Boiling tube or test tube, you can do either. Oh yeah, counting bubbles wrong as well. That's a good possible error. Well done. Okay, we're now moving on to respiration. So, why is mitochondria important in cells? We spoke about this question in actually the first topic of the first live stream that we did. What does acclimatize mean? Acclimatize just means get used to. So, because initially, obviously, if you've just taken a pondweed, which is in air, and put it in a test tube, which is full of liquid, it's going to produce loads of bubbles of just oxygen naturally. That isn't going to be the result of photosynthesis happening. Okay, so mitochondria. Mitochondria, well, cells require energy for metabolic functions, and therefore mitochondria is the site of aerobic respiration where energy is released. Thank you to everyone who said energy is released. Okay. Part one, I actually did in a live. If you want me to do part one in this live as well, I am more than happy to. We'll do a vote, but I don't see too much of a point because I actually just did it on Tuesday, so it's up to you guys. I'll do a vote after this. Okay, why does the heart rate increase during exercise? So this is a kind of a step-by-step question. So you need to think about how to answer this. So why does heart rate increase? Well, the heart rate increases to increase blood flow. Why do you want to increase blood flow? You want to increase blood flow because you want to supply more glucose for respiration so that you can create more energy. Now, if you want to supply more glucose for respiration, what else do you need for respiration? You also need oxygen. So if there's more blood pumping, it's pumping more glucose and oxygen. Okay, and then on top of that, why do we want the energy? Why do we want more energy for more work? Well, you want more energy so that the muscles can keep going consistently or even go at a faster rate if you're inclining with your exercise, if you're going faster. Another thing is to enable the muscles to contract faster. Another thing that's important as well is to remove lactic acid, because lactic acid is the product of anaerobic respiration, and that causes muscle cramps, and our body doesn't like it. We need to get that away from us as possible, as much as possible. So that's another reason why blood flow is pumped around the body. Okay, moving on to the next question. During exercise, the heart rate of smokers increases more than the heart rate of non smokers. Describe an investigation that would allow you to test this hypothesis. So, this is a very specific question, and they're asking you to design a practical. A lot of people didn't get over 2 out of 6 in this question. So, think about the way you would answer this. So, how the mark scheme has spoken about it is you're going to get 2 groups of people. One group are going to be smokers and the other group are non-smokers. Now, each group, we want to have certain control variables. So the mark scheme said have at least 5 people. Well done, Vixie, that's completely right. You're then going to get both groups to do the same exercise. That's another control variable. For the amount of time, which is another control variable. Then what you're going to do is you're going to ensure that the groups have the same health, the same age, the same gender. There's a lot of variables that could impact this that you need to make sure are at least very similar. What you're then going to do is you're going to measure the heart rate of each person before and after the exercise, and you need to ensure that you're measuring it via the same method. What then happens is you're then going to calculate the change in each person's heart and compare them. And that's what is enclosed in the mark scheme. You can speak about more points if you want. So it depends what answers you want to say. Okay, and now we're moving on to the next page. So this next page is actually the last slide of questions. So then we'll decide what to do after this. Okay. Oh, no. Here, metabolism. Explain why the number of phytoplankton are lower in the winter months. No, don't get confused. This question, there was context given to it before. So all you want to think about is, why is the number of this thing that lives in the water going to be lower in the winter months? Well, because there's a lower temperature of the water, that means that they actually have a slow metabolic rate, and therefore there's slower growth and there's less reproduction, because there's actually a correlation between the temperature of the area that you're living in and your metabolic rate. So perhaps if you come from a country, or if you have heritage from a country which has a higher temperature, your metabolism is going to be a little bit faster. Well, that's what science experiments have shown. And it's the same with looking at it in this phytoplankton. And therefore, because the slower metabolic rate, there's a lower rate of photosynthesis as the light intensity decreases as well, and many will die. Okay, why do the number of phytoplankton fall at the start of summer? So why would they fall at the start of a new season? Well, because the temperature begins to get higher, it causes a lot of the plants. You don't have to do phytoplankton. Don't get too confused. Phytoplankton was just an example of a question that they gave. So yeah, they don't expect you to know about that. So the warmer temperature was too high. All of the plants that had got used to the colder winter temperatures died because their metabolic enzymes started to denature. Okay, and what that then meant was that since their enzymes started to denature, a lot of the phytoplankton also began getting eaten by animals, as other animals were also experiencing the impact of the warmer temperatures. They were breeding more, and they were therefore more herbivores in number. Okay. Give 2 factors that affect a person's metabolic rate. Well, you can talk about a person's age. For people asking me to send the snap, I'm very confused. What snap do you want? Maybe that's just spam. Okay, a person's age, or you could talk about a person's gender. So those are 2 factors that affect a person's metabolic rate. The next one. One metabolic reaction is the formation of lipids. Give one other metabolic reaction in cells. So, respiration, photosynthesis, formation of urea, formation of glycogen. That's a more paper 2 question, so don't worry too much about that. Okay, the next one. Explain why the metabolic rate of an organism is greater than the metabolic rate of another organism, despite both being kept at a constant body temperature. So looking at what factors affect the metabolic rate, given that the temperature is exactly the same. So, you can talk about how it has a larger surface area to volume ratio, therefore it's more likely to lose heat. Because even though 2 organisms are both absorbing heat, one might lose heat at a faster rate. Another thing you can speak about is if it has a greater rate of respiration. If you are more likely to respire, then you are more likely to actually get rid of all of the heats that you've just had. And you actually require more heat to warm you up to the same temperature. Now, respiration is a large indicator of metabolism. So if they ask you a question talking about how metabolism, what things affect metabolism, you guys need to mention at least 1 point about respiration. Okay, and more heat needs to be generated as well. That's another point. This is not a predicted paper. No, it's not. Okay, so that is the end of going through the second half of the questions. If you wanted to go through the first half of the questions, go back to the live stream. Let me actually do a poll now to ask you guys whether you want to go over the first half of the topic's questions, or whether you want to go through a past paper. So I'll put a poll up on the community tab and you guys can answer it. And if you have any other suggestions, you can put it in the comments below. Okay. Okay, I've put the poll up now, so you guys should be able to answer it and tell me what you think. The past paper will probably be a triple paper, because most people are like that, are doing triple. But it's actually useful to both parties to do practice on everything. Okay, I've put the you don't have to answer on the chat. You can answer on the community tab for NOEL for GCSE, the poll. There is a poll now. Can you not see it? Some people have answered it already. Okay, so most people are saying go through a past paper. And if so, we'll talk about which past paper in a second. Thank you, whoever did that, put the link of the community tab. Okay, and just updating this. So most people, 64% have said go through a past paper. And okay, so it's going through a past paper seems to be the biggest choice. Would you want to do a past paper on combined higher or triple? Hardest past paper questions that have ever come up. That was more of what we were doing in the live stream. They were the harder questions. Unfortunately, they didn't have many hard questions. That's why I've handpicked these ones. So doing a past paper, there might be some easy questions, if that's okay with you guys. A lot of people are saying high, a lot of people are saying triple. So I'm not too sure which one to. Let's do another poll. I think that will be the best option. Okay, I'll do another poll asking, would you like to do a combined higher paper or triple paper? Okay, I've now put that poll up, so hopefully you can access that, and let's do that so we can see the exact number of people who want to do either. Thank you guys for all being here as well. It means a lot that you guys are still here. So yeah, please go to the YouTube community tab for NOEL for GCSE and answer the most recent poll, which is, would you like to do a combined higher past paper or a triple paper? You don't have to put it in the chat. There's a poll on the community tab. Okay, most people have said triple, so it seems like triple is going to be the winner, but it doesn't mean it's not applicable to combined higher. Most of the topics will cross over, and in fact, it's probably good for combined higher students because they're challenging themselves. Stop waffling. I'm speaking whilst I'm waiting for you guys to vote. Okay, it's triple it is. 123 people have voted. We're doing a triple paper. I'm sorry if that's disappointed some of you guys. I don't know which year you want to do. It depends which year you've done a lot of practice on. So GCSE AQA Biology Paper 1. There's the 2022 paper. Do you guys want to do that? 2024. That would be a good idea if we could access that. The 2023 paper is not available, so we can't do that. 2022 or below, I'm afraid. (2018) Okay, we're doing 2022 because that's the most closest to what everybody wants. So the 2022 Biology Paper 1. I mean, you know what? We'll probably get through a lot of papers. We're not only going to do this paper, we're going to do other ones as well. It's a shame that I can't write on the paper. That's really annoying. Okay. Well, let me try something else. Sorry, just deal with a couple of minutes of break whilst I just get the papers up. Okay. Seems to have gotten it up now, so I'm going to share the screen. Okay, I don't know why there's so many adverts on the left, but hopefully you can see this. So what you can do is you can start answering in the it's not even showing. You can start answering in the chat, and I'm unfortunately unable to write on this document, so it'll just be verbally going through it, and that might be better because we can actually. Oh, I'll show you the. Oh, there's no model answers. Yeah, that's what we'll have to do. Okay, let's go through the mark scheme of this as well. So, question 1, can you complete the table? It's starting off relatively simple, so let's see if you can complete the table. So the first one everybody is saying is a nucleus, so let's look at the answers. Yeah, the first one is a nucleus. People were getting confused earlier on, and this is a common misconception actually, that contains genetic information, yep. But people get confused with this last one, controls the movement of substances into and out of the cell. Now people think, okay, that's a nucleus because it's controlling the movements of the cell. But reality, if it's movements of substances into and outside of the cell, that's going to be the membrane, because the membrane controls what enters and exits the cell. The nucleus controls the cell at the same time, but it doesn't control what enters or exits it. So we've got our answer: nucleus. Mitochondria, the function, we've spoken about mitochondria a lot of different times. So what you want to say is it's a site of aerobic respiration. It's only a 1 marker, so that's all you have to put. Okay, moving on to the next one. Cells in potatoes are plant cells. Cells in potatoes do not contain chloroplasts. What is the function of chloroplasts? So what is the function of chloroplasts? I'm now going to go over the answers. So chloroplasts, wow, the mark scheme literally has one word for this question, which is photosynthesis. But I wouldn't recommend just putting one word. You want to try and maximize the possible chances of getting good marks. So you can put where photosynthesis occurs and energy is released through glucose, or glucose is created. Okay, name one type of cell in a potato plant that does not contain chloroplasts. Now, hopefully this should spring right out at you. The one that doesn't contain chloroplasts is root hair cells. Okay, they are in the soil, and therefore there's no point for them to contain chloroplasts because they're not going to get any sunlight. So instead, they get rid of that and they ensure they have enough space to absorb water and minerals. We've now got the osmosis practical being asked about. Okay. Thank you, Jack, for that £1. I hope everybody's listening or people finding this useful. I would recommend listening as opposed to typing in the chat, because yesterday, a lot of people who were listening, they realized that a lot of the questions actually that I spoke about came up in the exam. So it might be helpful to pay attention, but it's up to you guys, whatever you do. Okay, so a student investigated the effect of salt concentration on pieces of potato, and here they've given you the method for their osmosis practical. So they cut the potatoes, they record the masses, they add 150 centimeters cubed of the salt solution to the beaker, you put each potato piece into the same beaker. So what did we learn in our practical, or the practical that you would have learnt? There's 3 separate beakers, and you put each potato, a potato in each one, and then each of them have different solutions. So in this way, they're all in the same one, therefore they all are in the same salt solution. So clearly that is not an independent variable, it's not changing this time. Now, after 30 minutes, you then remove each potato piece, you dry the surface with a potato towel, and you then record the masses of the potato pieces. Okay. You then repeat these steps using different concentrations of salt solution. So that's how that's a slight adaptation of the practical that you would probably be familiar with. Okay, what is the independent variable in the investigation? I kind of told you the answer to this one already, but it is the concentration of the salt solution. Why? Because we know the masses is staying the same of the potatoes. We ensured that. The time that the potato is left in the salt solution, we're told that's the same. And the volume of the solution, it's 150 centimeters cubed, so that doesn't change either. Perfect. Let's move on to the next one. Why did the student dry the surface of each potato piece with a potato towel? So we actually went through the answer to this question in the first live stream that we did on Tuesday. So the answer to this is to make sure that the potato only has the mass that was measured, or we're talking about it removing excess water that is just on the surface of the potato. Okay, you don't want to squish it because you don't want excess, the water that's absorbed coming out, but any of the water that's on its surface is not absorbed via osmosis, it's just on the surface. So removing excess water, really good. Next one. The student calculated the percentage change in mass of each potato piece. For one potato piece, the starting mass was 2.5 grams. The end mass was 2.7 grams. Calculate the percentage change in mass of the potato piece. Now look how nice they are. They've even given you the equation. So hopefully you can try and calculate this. And let's see. So the individual steps. So what you want to do is you want to do 2.7 minus 2.5. Looking at the increase in mass, it's 0.2 grams. You're then going to do 0.2 grams over the starting mass. Sometimes people accidentally do 2.7 grams, but no, that's not the starting mass. The starting mass was 2.5. So you have 0.2 over 2.5 times 100, and you should get 8%. Well done for all of the people who've got 8%. Okay, and that's 2 marks. Moving on to the next one. The student used the results from each potato piece to calculate the mean percentage change in mass at each concentration. So you've got the different concentrations and the mean percentage change in mass. You can see this one is negative, and what does that suggest? That means that the mass actually decreased. So you're now asked to draw a graph. Obviously, you can't do this because it's digital, but labeling the axes, using suitable scales, plotting the data, and drawing a line of best fit. Now, the line of best fit is important. In terms of practicals, and this goes across biology, chemistry, and physics, knowing whether to put a straight line line of best fit or a curved line of best fit is simply, in my opinion, just about knowing the practicals really well. Now, if you know this practical really well, you should know that it's going to just be a straight line for this practical, but sometimes it's curved, like for a lot of the physics practicals, which we'll go through in the time of those ones, those live streams, they are curved a lot of the time. So this is a case where it's always a straight line, and that's the main issue there. Now, what concentration of salt solution, I told you they always ask this question, what concentration of salt solution was equal to the concentration of the salt solution inside the potato pieces? So you should get a graph, if you were to draw it, that kind of follows, I don't know if you can see my mouse, that'd be interesting to know, but it goes through the axis. It doesn't go through 0, 0, it goes through the axis and hits the x-axis at a certain point. So wherever it hits the x-axis, that is actually the concentration of it. Oh, it's not on full screen. I'm so sorry about that. It's not. Why is it doing that? I'm trying to max. Okay, yeah. There, I've maximized it a little bit. Okay. Moving on to the next one now. Explain why the potato pieces in the 0.4 moles per decimeter cubed salt solution decreased in mass. So when there's a decrease in mass in the osmosis practical, what ends up happening, or the reasoning for that, is because of the fact that water has moved out of the cell. So that's the first mark there. Water has moved out of the cell. You then want to say bioosmosis. And then your third mark is talking about why. Why has that happened? Well, we know that the solution in the potato is less concentrated than outside. So it means that the potato actually has a lot more water than outside. So what happens is, since osmosis, the water moves from an area of high concentration to low concentration, it moves from high concentration in the potato to low concentration outside. Okay, and that's the 3 marks. Moving on to the next one. Plant cells and fungal cells are similar in structure. So here you can see a fungal cell. Name one structure in figure 2 which is present in both plant cells and fungal cells, but it's not present in animal cells. Okay, so think about this. What is present in plant cells and fungal cells, but not in animal cells? Well, we know that we can see a cell wall here. So the first line is a cell membrane, and the outer line is a cell wall. And a cell wall isn't present in animal cells, but it is in plant and some fungal cells. So that's what you could say. Another thing you could also say is a large permanent vacuole. That's another option. Okay, let's now move on to the next topic. So well done for those who said vacuole as well. That's a really good answer. Which disease is caused by a fungus? We actually went through this in the live, so can you tell me which one is caused by a fungus? Let's see what the chat is saying. Yes, I think some people are saying rose black spot, and that is correct. Well done. Yeah, it's rose black spot. Perfect. Okay, moving on to the next one. Or just going through the other ones we spoke about malaria, that's a protist disease, measles, that's a virus disease, and gonorrhea, I think that's a bacterial disease. A fungal cell divides once every 90 minutes. How many times would this fungal cell divide in 24 hours? So here's a bit of math skills that are required. So what you want to do is calculate how many 90 minutes are in 24 hours. So what you can do is you do 24 hours times by 60 to see how many minutes are in 24 hours. And what is that? That is 1,440. And you then do 1,440 divided by 90 to see how many 90 minutes are in 24 hours. And you find out that that is 16. So 16 times, and therefore there's 16 divisions. Well done for those who said 16. Okay, moving on to the next part. Some types of fungal cells are grown to produce high-protein food. The high-protein food can be used to make meat-free burgers. Where is protein digested in the human digestive system? So let's go through the options and talk about which ones are which. So we have got, what are the options? You've got large intestine, liver, salivary glands, and stomach. So salivary glands, that starts digesting carbohydrates initially. The liver doesn't actually digest anything, it releases bile. So that's out of the question. So these two are out of the question. Now, the large intestine is responsible for digesting a lot of different things, but protein is not one of them, and therefore the answer is stomach. If you are even more aware, you know that there's actually pepsin. Yeah, well done for Arctic Blue, that's correct. Okay, now moving on to the next one. Another multiple choice. So you can see in 2022, they were quite generous with the multiple choice questions, weren't they? So, which chemical could be used to test if the burgers contain protein? So this is a food test question. Benedict's reagent, burette reagent, ethanol, or iodine solution? Well, let's think. Protein, for the test for protein, remember it turns it from, I think, blue to violet if it's there, or purple, and that is burette reagent. Okay, next question. This table shows some information about burgers made from meat and burgers made without meat. So you've got meat burgers and meat-free burgers. Evaluate the use of burgers made from meat compared with meat-free burgers in providing humans with a healthy, balanced diet. So evaluate, look at the pros and the cons. And now look at the data in the table. We're asked to talk about and discuss the different impacts of having a lot of cholesterol, of having a lot of fat, etc. So a couple of points that you can mention. You can say, look, meat-free burgers, they have a lot more fiber. And you want to use some statistics in your answer as well to show your application. So you're going to say meat-free burgers in terms of fiber, they have a lot more fiber. Meat burgers have a lot more protein. And then you can talk about what are not just it has more protein, you want to say what is protein needed for? Protein is needed for growth and repair. Therefore, the meat burgers are better or optimum or more efficient for if you're trying to improve the growth and repair, compared to meat-free burgers. However, meat-free burgers specialize in fiber, which means that the digestive system is going to be more healthy if you have meatless burgers. Now, meat burgers contain more fat, as you can see, 16 compared to 5.2. And don't be afraid to do some maths as well. You can find the difference and say it has this many more than meat-free burgers. And what is that? What are the consequences of having more fatty foods? Well, it could lead to chronic heart or coronary heart diseases, or having a heart attack, or leading to obesity, leading to diabetes. And make sure that you're speaking about type 2 diabetes, be specific, or type 1, whatever you're focusing on. Meat-free burgers are more ethical. Yeah, that's a good one as well. But in terms of the question, it's saying in providing humans with a healthy, balanced diet. So we're talking mainly about the different food groups here. Now you can talk even about carbohydrates. So you don't have to speak about all of them to gain the 6 marks. You want to make at least 3 comparisons. And then after those 3 comparisons, you want to do a bit of analysis and talk about what are the purposes of protein or fiber or fat or whatever you've spoken about. Ooh. Okay, so I hope that makes sense. We'll now go on to the next topic. A student prepared some onion cells. We know it's which practical is it? It's the microscopy practical. So you don't need to know actually what fiber is required for. You know it's helpful for digestion, but it's not too important. No, the graph isn't curved, it's actually a straight line. I think sometimes if the points are making a curved shape, you might want to draw a curved one, but normally most of the time for that practical, it's actually a straight line. Okay, moving on to the next one. So a student prepared some onion cells. The student viewed the onion cells using a light microscope. This is the method used. So you cut your onion into pieces, as we said, you peel off a thin layer of onion epidermis, which is just a thin layer of skins, and it's often the bit of onion that you're peeling off is a very see-through one on the edge, using a sharp knife or a scalpel. You're then going to place the onion epidermis onto a microscope slide in a single flat layer. You're going to add 3 drops of iodine solution, lower a cover slip at an angle, and then place the slide on the stage of the microscope. So you can see here that the method that they spoke about is only talking about the preparation stages. Okay, there's obviously other stages which are also quite important for this, such as actually using the microscope, using the objective lens, the coarse focusing lens, moving the stage up, or if it's one of those light microscopes where you have a mirror and you have your light reflecting from a mirror. Those are the old style microscopes, but a lot of people have the newer ones now. Okay, let's look at this now. Table 4 shows a risk assessment for the experiment. Can you complete the risk assessment for this practical? So not many people consider the microscope practical to have a lot of risks, which is why they're trying to ask the more unexpected parts of each practical. So what we're then going to say for this is iodine solution is an irritant. The risk, it may cause an allergic reaction to the skin, and therefore, what are the ways to prevent that from happening or to minimize the risk? Well, you can wash your skin immediately, but that's obviously after contact. So to prevent it, you want to wear gloves, or you want to, that's the main option there, to wear gloves, or maybe wear a lab coat as well. Okay, another one. Sharp knife. What is the risk of a sharp knife? Well, it may cut you or it may cut someone else. That's the risk. Now, plans to minimize that risk, you can say cut away from the body, use a chopping board, keep your fingers away from the blade, facing away. You probably learned this stuff in food tech if you did that. Or any sort of method the mark scheme allows for talking about how to use a knife safely. Okay, next one. Give a reason for each of the following steps in this method. So we're really breaking down the method now. So why is a thin layer of onion epidermis used? This is only 1 mark. Each of these 3 points, you have to just use a 1 mark explanation. So why is a thin layer used? Well, it's to help see the individual cells, because that's what you're trying to do. That's the purpose of this experiment, to see individual cells. So that's what you are aiming to do. Your next one. Iodine solution is added to the onion epidermis. What is the point of that? Well, the purpose of that is to stain parts of the cell. If you stain parts of the cell, it's more clear, and that's the point of it. You're trying to increase as much as you can the clarity of it. So you'll do that in whatever way you can. Okay, and the next one, which is actually why a lot of people got confused in this question here. The cover slip is lowered onto the onion epidermis at an angle. Why is it done at an angle? Well, one of the key reasons is to prevent air bubbles. Or the mark scheme also accepts reducing the air bubbles there as well. Okay, so moving on to the next question. I hope you're finding this as useful as what we were doing before. Okay. A lot of people are joining in to the chat, which is really nice to see that you guys are putting a lot of effort into your exams. Okay, so here we've got what the student saw under the microscope. It's a picture, it's a photograph. Now so the real length of the cell is 4.8 centimeters. Calculate the length of the cell in the image is 4.8 centimeters. Calculate the real length of the cell and give your answer in micrometers. So there's a couple of skills tested here, your knowledge on the magnification formula and also your knowledge on conversion. So the conversion is right at the end. So let's start with the magnification formula. Yeah, this video or this live stream will be uploaded for you guys to access if you want to go back to it. So magnification, the formula is the size of the image over the size of the real object. Okay, that is what magnification, that's a formula of it. So in fact, though, we're not trying to find the magnification in this question. You're asked to find the real length, i.e. the size of the real object. So you can rearrange the equation and you say the size of the real object is the size of the image size over the magnification. So real object is image over magnification. And that is just 2 points there already scored, just by writing down the equation and then rearranging the equation. So hopefully you can see the importance of writing the equation down. Okay, the next step, once you've rearranged it, is to substitute your values in. So you substitute 4.8 in. We know that's the size of the image. And your magnification, we're told here, is times 400. It's often put up so you can obviously miss it quite easily, but it's also in the diagram here. So you then do 4.8 divided by 400, and your answer should be 0.012. Now, that is centimeters, so you need to convert it to micrometers. And the conversion from centimeters to micrometers is to multiply by 1,000. So you go from 0.012 to 120 micrometers. Okay, and that was 5 marks. So look at that multi-step problem. Let's now move on to the next one. Okay, so here's the student's drawing of onion cells. Okay, give 2 ways that the student could improve the drawing, because at the end of this practical, a lot of people forget that the purpose of the practical is at the end, you draw out what you see, because you want to then draw the magnification, how much everything's been magnified by, etc. So what's important to then say there is what is wrong with the diagram. People are saying it already, there's no shading. You're not allowed to shade in the diagram. You're not shading the nucleus. You're just drawing. Another key thing is you want clear, unbroken lines. There's loads of possible things you could say to improve this. A thing that they missed is they didn't include the magnification or the scale. They drew overlapping cells, and that's not possible physically. Obviously, there's one cell occupying one space, so that shouldn't happen either. And the main thing is clear, unbroken lines. Also, there's no labeling as well. There's the onion cells, but then there's no labeling of the subcellular structures. So those are the things you could say. Onion cells can be seen using an electron microscope. Give 2 ways onion cells would look different when seeing an electron microscope. So here you're using it's an application of your understanding of what is the difference between a light and electron microscope. You're applying that to the idea of how cells would look different. So different ways it would look different. You can speak about you can obviously say it would look more magnified. The cell would have much more detail. You could say you can see more subcellular structures, and you can say there's a higher resolution as well. You can see the finer details more clearly. So any of those comments about magnification, resolution, or subcellular structures would be ample for those marks. Okay, moving on to the next question. Here are some plant defenses. Identify whether each defense is a chemical response or a physical response. Okay, so you've got a thick waxy layer on the leaf surface. Is that chemical or is that physical? That is actually a physical one, because it's I don't know how to explain the differences between physical and chemical, but there's not many chemicals involved in that. There's no release of anything chemical. The next one, berries that are poisonous. This is definitely chemical because the poisonous chemicals are the things that lead to this happening. Bark on trees that falls off, that is a physical process. So hopefully it's a bit more intuitive. Well done, guys. You got it right in the mark scheme, in the chat. Okay. Moving on to the next one. Mimicry is a mechanical adaptation seen in both plants and animals. So we can see images of two insects. Hornets are insects that sting other animals and cause pain. Hornet moths do not sting other animals. Suggest how mimicry helps the hornet moth survive. This is simply one marker, but there's a lot of thinking that goes towards this. So firstly, you've got to understand that the idea that they act as a the idea that they have a stinging property, that is a good property for protecting themselves as a defense mechanism. So you've got to firstly understand that. Secondly, then you've got to understand that the hornet moth, by mimicking it, is therefore kind of impersonating it and suggesting that it has that property. So therefore, it's using the idea that animals are tricked or deceived by it and avoid eating it to avoid the pain. That's what the mark scheme would suggest. Okay, moving on to the next topic. Adult hornet moths lay eggs that hatch into larvae. So you can see the larvae in the figure. The larvae of the hornet moth live inside the roots of trees. They also use the tree roots as a source of food, and they cause damage to the tree roots. So they're quite the annoying pests. Explain why a tree might die if the roots of the tree are damaged. Now, this is a 6 marker, and it's well worth going over this in detail. There's a lot of possible impacts of the hornet moths that could cause the tree to die because of the roots. So if we're looking at the roots, what are the different cells in the roots that have a massive impact? Well, we've got the root hair cells. And the root hair cells are not just responsible for the absorption of water via osmosis, but also talking about the absorption of mineral ions. Now, this question is specific. They want you to have named exactly what mineral ions and what are the purposes of those mineral ions. So let's begin with less absorption of water. If there's less absorption of water, this means that there's a lower rate of photosynthesis because we know water is a reactant in photosynthesis. If there's a lower amount of photosynthesis, there's less glucose produced. The plant is unable to respire as much as possible, and it's less likely to grow. On top of that, there's fewer amino acids produced to make new proteins. There's less cellulose produced, so there's fewer cell walls being made. Another thing is that the cell loses turgidity as well because the water ensures that the cells are turgid. They have enough water to make them very strong. If there's less water, they become flaccid and very easy to attack. Now, another thing about water is that it helps to cool the plant down, and that's really important as well for hot days, depending on the climate that the plant exists in. Now, less absorption of named ions and minerals. So, talking about nitrate ions to begin with, which I see some of you speaking about in the chat. So, if there's less nitrate ions, that means that there's fewer proteins. And if there's fewer proteins, there's less growth. Now, if you're talking about magnesium ions, if there's fewer magnesium ions, we know that magnesium ions create chlorophyll. So, therefore, less chlorophyll is produced. If there's less chlorophyll produced, that's another way of reducing the rate of photosynthesis. Yep, you can talk about the tree falling over. That is perfectly a good answer. Okay, now you can talk, and another thing to talk about is the damage to the phloem and damage to the xylem. as well. So the damage to the phloem means that there's less transport of sugars, which are really important for storage and for respiration, for energy release. There's damage to the xylem because if there's less water produced, if there's less water transported to the cells, then the xylem will actually get damaged as well, because the xylem needs water to survive, as do most cells, living cells, and therefore there's fewer nitrates that reach the cells. If the xylem is not working and damaged, then there's fewer ions that reach cells, fewer magnesium ions, so again, less chlorophyll is produced. And the point about it falling over, you can talk about less anchorage that is in the mark scheme, so how the roots anchor the tree in the ground. Okay, let's now move on to the next question. The larvae of the hornet moth form when fertilized eggs divide by mitosis. So this is a really nice topic link you can see. Describe how mitosis produces two genetically identical cells. So this is a 4-marker, and it's relatively straightforward as long as you know the process. So remember, the first stage is a longer stage. It's when all of the genetic material and the subcellular structures are duplicated. It's important that you apply that you don't just say they increase in number, you say something like they're doubled or they're replicated or there's twice the number, because we know that they then split into identical cells. So in order to do that, they need to have double the number. This is a triple paper from 2022. Okay, the next stage is then the replicated chromosomes are pulled apart from each other onto either end of the cell. And then finally, the final stage is that the cytoplasm and the cell membrane divide. And that forms two identical daughter cells. And the set of chromosomes in either of the daughter cells are identical. So any of those points you mention, and you'll be on to 4 marks. Yes, some people know the exact names for each of the mitosis processes, and that's actually not necessary. It's not required for you to know them. It's actually good if you do, because you can mention them in the exam and impress the examiner. But if you don't, I wouldn't tell you to have to memorize them. Okay. The cells which are first formed from the fertilized eggs of the hornet moth are stem cells. Name the process by which these stem cells then form specialized cells. So they don't want you to say specialization. A tip that they're telling you that is because they've spoken about specialized here, so it's clearly differentiation. Okay, question 5. Water and carbon dioxide are exchanged between leaves and the atmosphere through pores called stomata. Name the cells that control the opening and the closing of the stomata. So these are the cells that literally are the only reason why the stomata are there, and they are the guard cells. Perfect. I can see people already saying it in the chat. Well done. Water moves through a plant in the transpiration stream. Describe 2 differences between the transpiration stream and translocation. So this is actually a question we went through in the live stream. So hopefully you remember the answers. So remember, you can say anything. There's lots of differences. You can say transpiration involves xylem tissue and translocation involves phloem tissue. Or you can say transpiration transports water and mineral ions. Translocation transports dissolved sugars. You can say the transpiration stream moves substances upwards into a single direction flow, unidirectional, whereas translocation is moving upwards and downwards, so it's bidirectionally. Well done, guys, in the chat. It seems like you are paying attention, answering the questions really well. Next up. Which environmental conditions would cause the rate of transpiration to be greatest in a plant? Now, a lot of people neglect learning about the factors that affect the rate of transpiration. So let's see if you can answer this. Cold with low humidity, cold with high humidity, warm with low humidity, or warm with high humidity. So, talking about the temperature. Now, most people get the temperature part of it right. The transpiration is going to be faster if it's warmer, because if a plant is hotter, there's more sunlight, the plant wants to undergo more photosynthesis, so more water is dragged up, the transpiration rate increases. However, the humidity people often get confused about. So humidity is literally how much water there is in the air. If there's high humidity in the air, that means that the water is less likely to diffuse from the stomata out into the air. Why is the water less likely to diffuse? Because there's not a very large concentration gradient. There's not a very large difference between the two areas in terms of concentration. So you want low humidity. You want it to be very dry conditions so that therefore a lot more water is going to come out and the transpiration rate will increase. So everybody who said warm with low humidity is absolutely correct. Well done. Moving on to the next one. Here, this graph shows information about the mean width of the stomata in a plant. Now, I think we actually went through a question about this, so you guys should be aware of that. So the changes, so we did actually answer this question, this exact question in the live stream, so we're going to skip this one. Okay. The changes in the mean width of the stomata in low atmospheric carbon dioxide are different from the changes in normal conditions. Explain how the difference helps the plant to survive in low atmospheric carbon dioxide. So just looking at the graph to see what is actually the difference. Low atmospheric carbon dioxide, you can see that the stomata opens greater. So the stomata opens wider when there's not much carbon dioxide in the atmosphere. Okay, I can see some of the chat, but I can't see all of them, by the way, because it is going quite at a fast rate. Okay, so let's go back to the question. Explain how the difference helps the plants to survive in low atmospheric carbon dioxide conditions. So the stomata, the first mark is given for acknowledging that the stomata open wider and for a longer period of time as well. So acknowledge that the stomata isn't just open wider. You can see that the width of the graph is greater. It's open for a longer period of the day. So it's doing two different things to help increase the amount of carbon dioxide. So it allows the plant to take in more carbon dioxide for photosynthesis. The mark scheme does say that you don't have to necessarily mention the time, you just have to mention it opening wider, and you will get the 2 marks. Okay, moving on to the next one. This table shows information about 5 different organisms. Calculate the value X in the table, the surface area to volume ratio. So hopefully this is just testing your understanding of being able to do ratios. So what you want to do is put the 2 numbers together and then divide by the smaller number. And that should help you get the surface area to volume ratio. So you will do 9.96 times 10 to the power minus 3 divided by 1.35 times 10 to the minus 4. You'll get a number like 73.77. And they said to the nearest whole number, so just round that up to 74. And therefore the ratio is 74:1. So I hope that makes sense. Well done, Sonic8. I can see you got the correct answer. Okay, next question. What is the relationship between the size of an organism and its surface area? And it says use table 6. Now, when it's told you to use it, you have to use the data. So let's see how we can use the data above. Well, you can see that as the size of the organism increases, the surface area to volume ratio decreases. Okay, so it gets smaller and smaller. And you can use the facts and figures from that to support your answer if you want. Okay, next. Organism B exchanges gases with the environment directly through its skin. Organism D exchanges gases with the environment through its respiratory system. Explain why organism D requires a respiratory system, but organism B doesn't. So we've got B and we've got D here. Now notice they are in different rows. D, you can see, has a much larger volume, and therefore the surface area to volume ratio is almost 100 times less. That means that because it's got such a low surface area to volume ratio, it can't just rely on transport through its skin. What it has to do, therefore, is then get adaptations in order to better increase the gas exchange. So what you want to put in terms of the mark scheme is D has a smaller surface area to volume ratio, so the diffusion distance is too large. Yeah, okay, that's a good way of explaining literally why the surface area to volume ratio, a small one means that you're not going to have a lot of good gas exchange because the diffusion distance is too large. So that's what it says in the mark scheme. Okay. So this table shows information about organisms D and E. We're told the metabolic rate in arbitrary units, which just means units that they've figured out that doesn't really matter. So 890 and 75. So effectively what we're told from this is that D has a much larger metabolic rate. Now going back to it, E is actually a larger organism. So D has a larger metabolic rate. Now let's see what we can do with this data. Organisms D and E both keep a constant body temperature. They are warm-blooded. Explain why the metabolic rate of organism D is greater than the metabolic rate of organism E, and use information from the tables. So, organism D has a larger surface area to volume ratio, as we've said, because it's higher in the table than E. So because it has a larger surface area to volume ratio, that means that it will actually lose heat more quickly than E. Okay, so it's more likely to lose heat because more things are being exchanged through the skin. And therefore, D requires a greater rate of respiration because of the fact that a lot of its substances get lost through the skin as well. So this is quite a difficult question because they're kind of asking you to consider the reverse process of things. Instead of things entering your body, rather things escaping your body as well. Okay, so going back to the question, explain why the metabolic rate of organism D is greater. Well, you want to say that because organism D is more likely to lose out all of the substances that it's getting. It needs to be able to generate a greater force in order to retain that. Therefore, its metabolic rate needs to be higher. So I hope that makes sense. There's a bit of a broken explanation. Okay, let's now move on to the next question. We should be getting towards the end of this paper. It seems endless. Okay, organism D and organism E both have alveoli in the lungs and villi in the small intestine. Figure 8 shows some alveoli and some villi. Describe how the alveoli and villi are adapted to increase absorption. So this should be quite a nice question if you were to get this. So thinking about the different parts here, and what helps even more is that they gave you a diagram, so you can literally take all of the adaptations. Okay. I realize that it's gone over 2 hours, but hopefully some people want me to continue. So if you don't want to continue, you can leave the live stream, but it's up to you however you want to deal with it. But thank you. I value those who are staying on the live stream. I hope that you are finding this helpful. Okay, let's continue. So the alveoli and the villi are adapted to increase absorption. So you can see that the alveoli and the villi have very thin walls, which means it allows for rapid exchange of substances. And you can even list which substances. So for alveoli, we're talking about the oxygen and carbon dioxide. And with villi, we're talking about the things like minerals and things that we want to absorb from food. Okay, another thing is the blood supply, the large network of capillaries. That is really integral because it then means that there's, again, rapid exchange of surfaces. And now another point that you can make is that the villi have microvilli, which increase the surface area. They also have access to a lot of mitochondria, villi cells, so that helps with the active transport, because a lot of the villi do is active transport. Okay, so now moving on to the next question. Hopefully it's going to be the last questions for this paper, I believe. Okay, HIV virus, it's a pathogen. Give one way that HIV can spread from one person to another. Again, this is a relatively straightforward one. You can say from sexual contact, from intercourse, or from exchange of bodily fluids. Next, we've got a table which shows information about new cases of HIV diagnosed in the UK. So you've got the different years from 2010 to 2018, the number of new HIV cases in women, and the number of new HIV cases in men. Describe the trends shown in the table between 2010 and 2018. So what are the trends? Well, we can see that the number of cases in women decreases over time, and but it doesn't just decrease because it decreases, then it increases in 2014, and then it decreases again. Now, don't think that's not a nice smooth trend, so you can't talk about it. You can most definitely talk about that. It kind of fluctuates. Whereas with men, every year, in 2014 it increases, but it mostly decreases in every other year. Okay, and let's look at the next question. Suggest one reason for the change in the number of new HIV cases between 2014 and 2018. So i.e. we know that between 2014 and 2018, the number of HIV cases actually reduced. So what caused that to happen? Well, you could say better education, education of prevention methods of HIV. You can talk about contraception and things like condoms being more widely available. Don't just talk generally about contraception, because you want to talk about condoms specifically because they are the barrier method. You can talk about there being better drugs to prevent HIV infection. You can talk about there being better testing and identification of HIV as well. Yeah, you don't want to speak about vaccination, because vaccination won't impact the number of it won't necessarily impact the number of cases, because HIV was a thing where there isn't actually a vaccine for. That's why you can unfortunately die from HIV. There's immunosuppressants which help to reduce the pain, but there's no actual vaccine for that. Okay. Calculate the ratio of the new cases of HIV in women to the new cases of HIV in men in 2018. Give your answer to 3 significant figures. So again, going to 2018 and using the same method of ratios that we looked at in the surface area to volume question. So what you want to do is you want to do 1,200 and actually, Actually, no, this is a slightly trick question. So calculate the ratio of new cases of HIV in women to new cases of HIV in men. So what you might want to do instead is that you actually are going to divide the smaller number by the larger number. So you're going to divide 242 by 1,288. Yes. So it's not always the bigger number divided by the smaller number. And you should get 0.188 colon 1. Okay. Moving on to the next one. We missed a question. Where's 7.5? Here. In the UK population, the total number of women is greater than the total number of men. The data in the table is used to compare the proportions of new cases of HIV in the population for men and women. Suggest how the data could be presented differently so that a more valid comparison can be made. So let's go back to the table to see how the data can be presented differently so that there's a better comparison. Well, you can say that calculating the numbers as percentages, rather, because then you can that's a more fair way of comparing the proportions rather than just random figures, or giving the number as per 100,000 people. So again, any method of trying to calculate it as a percentage or dividing by a certain number. But if you just say as proportions, you won't get the mark. Okay. And 7.6. Scientists have been working on a way to produce a vaccine for HIV. Explain how a vaccine for HIV could work to prevent a person developing the HIV infection. So here we're just bringing together our knowledge about how vaccines work and just applying it to the case study of HIV. So as we said, you put in the inactive virus, in this case the HIV, you inject it into the bloodstream. Your white blood cells, specifically your lymphocytes, then are stimulated to produce antibodies against this inactive virus. Because it's inactive, you do not feel many symptoms as much as if you had the active version of it. If you're infected with HIV, specific antibodies are produced quickly, therefore via the memory cells, and it will then work. Okay. Moving on to the next one. So this question actually hints at the answer to the previous one, where a vaccine therefore hasn't been produced, and therefore you can't put vaccine as your answer to the previous question. Okay, a person with late-stage HIV infection has AIDS. Scientists have produced monoclonal antibodies for HIV. The monoclonal antibodies can prevent a person infected with HIV from developing AIDS. So describe how the monoclonal antibody for HIV can be produced. So again, we're talking about how that works. So how monoclonal antibodies are produced and how they work. So again, we've a similar thing to what we were speaking about. So the antigen, in this case, the HIV, is injected into the mouse. The mouse's lymphocytes is then stimulated to produce loads of antibodies. The lymphocytes are then extracted and collected. You then combine the lymphocytes with a tumor cell to create a hybridoma, and the hybridoma is then cloned to create many cells that produce the antibody. We already spoke about this process, so just go back to that portion of the live if you need a reminder. This figure shows how HIV enters a human cell. So here you can see the complementary binding sort of in the diagram. Suggest how the monoclonal antibody for HIV helps to prevent a person infected with HIV from developing AIDS. Effectively, so how does it work? And this is a 3 marker. So looking at possible ways. So the monoclonal antibody, as we said about the complementary lock and key theory, so it's the antibody is a complementary shape or specific shape to the HIV antigen. The monoclonal antibodies then attach to the HIV antigens. So the HIV cannot actually bind to the human cell, or the HIV genetic material cannot enter the human cell, and that's how it prevents it from working. Okay. Thank you, Joanna, for that. If that's towards me, I'm glad you're finding this useful. So, and that is the end of this paper. So well done. We made it finally through the 2022 paper for triple, that was. So what would you like to do now? Would you like to go over another paper, another triple paper, or what would you like to do? Some people are saying do combined. I'll probably do another combined paper for 15 minutes and then... Yeah, I'll probably end this video at 8:35 maybe. Final tips. Should I do some final tips? Because I think all combined. Poll. I think a poll is a good idea. Let me do a YouTube poll. I don't actually have predictions for the paper. I haven't done research into what possible predictions. I always recommend just revising everything. That's what I did and still managed to get grade 9s. So I don't think predictions are necessary. I know a lot of people are using them and that's totally fine as well. But I recommend just revising everything. As you can see, the paper is quite long. So a lot of topics could come up. So let me do a poll right now to see what would you guys like to do. Okay, I've put the poll up now. So you guys should be able to answer it. It's on the community tab. By the way, the final GCSE tips I'll release in a short or a YouTube TikTok or a YouTube short or a TikTok or any of those things just before the exam. Okay, so if you can go into the YouTube poll in the community tab, it'll be great. And just to get some feedback as what you guys would like to do, is it a combined paper, triple paper, or go through final tips? So it seems quite close right now. The poll is at the community tab on the YouTube. Oh, okay, it's very close for combined and triple. Let's see if someone can More people are saying combined now. Okay, that's interesting. Okay, let's do a combined paper. I think the combined students have had to wait and sacrifice during the triple, so let's help them out now. Okay, so let's do the combined paper. Okay. And it'll still be useful for those who are doing triple as well. It should still be beneficial for you guys. Okay. Just bear with me 1 second as I get a combined paper up. Okay, so we'll do the most recent one. We'll do the 2022 combined paper. You want to do 2019? Okay, I'll change it to 2019. That's fine. Okay, let's do the 2019 paper and then we'll do some final biology tips afterwards, okay? So let's look at the questions. This is going to be a much shorter paper to go through, so let's speed through this and do really well. Okay. So, figure 1 shows a human heart. Which blood vessel carries deoxygenated blood away from the heart to the lungs? Now, if you're unsure about the way that the heart works, I really recommend you go to the first biology live as we explained it there. But the answer is B, as people in the chat have been saying. Yeah, it's B, correct. The natural resting heart rate is controlled by a group of cells that act as a pacemaker. Where in the heart are the pacemaker cells found? This is a good practice for you guys. So is it found in the left atrium, the left ventricle, the right atrium, or the right ventricle? Can you answer in A, B, C, or D? Okay, or people answering just straight up. So yeah, it's in the right atrium. Okay, so just remember that the right atrium has the pacemaker in it. That's one main difference between the right atrium and left atrium. Okay, some people may be treated with a drug to slow their heart rate. Digitalis is a drug that slows the heart rate. Where does it originate from? Oh, wow. So this is interesting because for triple, you're not required to know where it comes from, but maybe combined you are. Or maybe it says in the specification. Okay, so it comes from foxgloves, which we spoke about. It wouldn't originate from bacteria, so that's immediately cancelled out, or mold. And we know that willow gives rise to aspirin. So maybe it was just a method of taking out the options that it can't be, rather than you expecting to know it. Lovely. Okay, let's move on to the next one. Beta blockers are another type of drug that slows the heart rate. In this table, it shows information for people who do not take beta blockers and for people who do. Stroke volume is the volume of blood pumped out of the heart every time it beats. Cardiac output is the total volume of blood pumped out of the heart every minute. Calculate the stroke volume X in table 1. Use this equation. So this is literally the case of plugging in the values and using the equation. This is the 2019 paper for those asking. So the answer, so what you would do is you would do X, the value is 2,800 divided by 52. And you just work that out, and then you're asked to put it to two significant figures. So 53.846153 to two significant figures is just 54 centimeters. Okay, so remember it's two significant figures. This is combined. Okay, moving on to the next one. Some people who take beta blockers get out of breath when they exercise. Explain why beta blockers can have this effect during exercise. You should refer to information given in table 1. Now, this is a 6 marker, so you definitely want to think about referring back to the table. So, looking at the effects of exercise and how beta blockers cause people to get out of breath. So during exercise, our body needs to transfer more energy to our muscle cells. So our blood is pumping, our heart is pumping more so that our blood can get around and transfer a lot of the oxygen to the muscle cells, and therefore to get energy. Therefore, in order for that to happen, the rate of respiration increases so you can inhale more oxygen. Okay, and as you're inhaling more oxygen, this is increasing the amount of glucose that's being oxidized, and you're getting out more energy. Now, the effect of beta blockers, what beta blockers do is they reduce the heart rate. So if they're reducing the heart rate, you're effectively artificially preventing or limiting or putting a cap on the amount of oxygen you can get when you're exercising. So your heart can't supply enough oxygen at a fast enough rate, and therefore your muscles will start to tire out. You'll get lactic acid, and your breathing rate will therefore increase really a lot, so that you can increase the amount of oxygen absorbed, because we know that the heart rate cannot be increased, so we're going to resort to increasing the breathing rate. But again, increasing the breathing rate isn't fully compensating when you can increase the breathing rate and the heart rate. I want to write the answers, but unfortunately the pens and the typing mechanism for this PDF isn't allowed. So I'm sorry about that. I was going to do that. But it's a good idea, Zahran. Okay. And yeah, so that answers that 6 marker there. Next one. Name the enzyme that digests starch in the human digestive system. So what enzyme is that? Can you put it into the chat? So I don't see any answers coming up. Amylase. Yeah, it's amylase. That's the answer. Perfect. Correct. Okay, moving on. A student sets up a model to represent the digestion and absorption of food molecules in the digestive system. So the test tube, you've got the water, the partially permeable tubing, starch and enzyme solution, and a water bath. So this is the method. You fill a test tube with water at 37 degrees Celsius. You test the water for starch and for sugar. You then mix together the starch and enzyme solution, and you immediately test it for starch and for sugar. You then fill some partially permeable tubing with starch and enzyme mixture. So you put that inside here. And you then seal the tube and place it inside the test tube of water. You place the test tube in a water bath to heat it up at 37 degrees Celsius, body's temperature. After 30 minutes, you test the mixture inside the partially permeable tubing and test the water in the test tube for starch and for sugar. This is not foundation, this is combined higher. So suggest which parts of the body the partially permeable tubing and the water in the test tube represent. So the partially permeable tubing represents the small intestine, but you could also say the stomach, because you're basically asking where in the body is starch absorbed. And we know that that's mostly in the small intestine, but also in the stomach as well. Then you want to say water in the test tube. So what is that supposed to represent? That is supposed to represent our blood. Okay. So moving on to the next question. We've got results now. I would say try to avoid spending time trying to find paper leaks. Instead, focus your time on revision. It's just a waste of time. And unfortunately, a lot of it is fake anyway, so you'll just be confusing yourself. I would recommend just revising, and I think you'll do absolutely fine that way too. Okay, so here are the results. You've got the 4 different tests, the description of the liquids, results of the starch test, and results of the sugar test. Name the reagents used to test for starch and for sugar. So, you can see people already answering. So the test for starch is iodine, and the test for sugar is Benedict. Okay, now moving on to the next question. Why was there no sugar present in test tube 1? So what we've got is a mixture inside the tubing at the start. So why was the result of the sugar test nothing here, but then in 3 and 4 it was there? Well, the only difference between the test tubes is the amount of time. So initially at the start, what happened was there was not enough time for the enzyme to work. That is what they want you to talk about. Explain the results for test 3. So test 3, the mixture inside the tubing after 30 minutes, the result of the starch test and the sugar test both were positive. So why is this? Well, the enzyme that was working the enzyme finally was able to break down the starch, and it broke down the starch and formed sugar. However, it doesn't mean that all of the starch was broken down. So there was still some starch left, and therefore there's also sugar, because that's what some of them had been converted into. So during that period, there's starch and sugar because the enzyme hadn't completed finishing its work. Okay, explain the results for test 4. Water in the test tube after 30 minutes. So at this point, we can then say that the enzyme had completed its work right at the end. All of the starch had been converted into sugar. And what also happened was interesting is because we're talking about the water and how it's got to the water is because the sugar molecules formed are small enough to pass through the tubing and eventually get into the water. So that's why we're breaking down the big molecules of starch into smaller sugar molecules so that it can eventually go into the water, which is supposed to model the blood. I can't close the advert because if I close this, wait. The advert doesn't close, unfortunately. I'm so sorry. I can pause it, but if I exit it, a new advert will just come, so it's not really going to be useful. Okay. Next one. A student used a potometer to investigate the rate of water uptake in a plant shoot. See, okay, I think that some of these questions are actually harder than the triple questions, so I think these are quite useful. for triple students to use. Now, going over this, as the chute takes in water, the air bubble moves. The rate of water uptake is the distance the air bubble moves, and that's measured by this ruler, as you can see. So if there's a lot of air uptake, it will pull the air bubble forward, and therefore that will work. So here are the conditions used: moist, dry, and dry. After the investigation A, the air bubble had moved partway along the capillary tube. Suggest how the student moved the air bubble back to 0 millimeters from the start of the investigation B. So how was that possible? How did they go about that? Well, let's look at the apparatus. What have we got here? Well, we've got our water, we've got our capillary tube, we've got our tube. Oh, look here, we've got a tap. So with this tap, you can then pour the reservoir of water. This will then push through, and all air bubbles will be getting rid of. Exactly. Well done, guys, for saying tap. Next, capillary tubing is very narrow. Explain why the narrow tubing was used. Well, because it's very narrow, it allows the air bubbles to pass through and move. And the resolution is improved, and also it means that it's very easy to count individual air bubbles rather than grouping up together and forming a large air bubble, because that could impact the ability for the distance it could move. So it's helping the control variables as well. Okay, now we've got a graph which shows the results. So for the investigation, the cross-sectional area of the capillary tube was 0.8 millimeters squared. Calculate the rate of water uptake for the investigation, millimeters cubed per minute. So all you want to do is, let's look at our values. So you've got roughly 66 millimeters of air bubble position here, and that is done in 5 seconds. So you can calculate the gradient here, the change in the y variable, the change in this, so 66, over the change in the x variable, 5 minutes. So 66 divided by 5, and you get 13.2. Okay. But we don't just stop there, because we then multiply this value by 0.8, because we're told that the cross-sectional area is 0.8. And you multiply 13.2 by 0.8, and you should get 10.56. Okay, moving on to the next question. You've got the results for investigation B. So plot the data and draw a line of best fit and label the line. So that's on the same graph. Investigation C was carried out in dry air at 25 degrees Celsius. Draw another line to show what you would expect. So, since it's more drier conditions, how is that going to impact the line? Well, it's going to be a more steeper line, so it's going to be higher. There's the position of the air bubble will go further in less time because of the dry conditions. The investigations were carried out in daylight. The air bubble would not move if the investigations were done in the dark. Explain why. Well, obviously, the photosynthesis wouldn't take place if there was not enough light. And because the photosynthesis wouldn't take place, that means that the stomata end up closing. Because remember, the stomata closes at dark times, and therefore no transpiration is going to take place because there's no way for the transpirational pool to even occur. Okay. Pathogens are microorganisms that cause infectious diseases. What type of pathogen causes malaria? I told you guys this in the live stream if you've been paying attention. So let's see if you can get this one. Okay, and well done. Everybody has put protest, which is completely correct. Next, give 2 methods used to prevent people from catching malaria. Oh my gosh, we even went through this. So it is the insecticide nets, the mosquito nets, insect repellents, vaccination, anti-malaria tablets, any of those things that we already spoke about. Okay, moving on. Describe two differences between a bacterial cell and a eukaryotic cell. So we spoke about this in the live stream from Tuesday. So bacterial cells, they do not have a nucleus, or you can say their genetic material is enclosed in a DNA loop or plasmids. A protist is effectively a pathogen that's transmitted via a vector. For example, in the case of malaria, the vector is the mosquitoes. So anything that's transmitted by another organism is what you call a protist. I'm going to do physics and chemistry. I'm actually doing live streams for most GCSE subjects. So I hope that you guys. Okay, so going back to this. Two differences between a bacterial cell. So we spoke about the genetic material, and you can also say, it's weird because on the mark scheme it says that you can say that the bacterial cell is smaller, but in most cases in triple, you're not really advised to talk about the difference in size because it's less scientific. Moving on. A scientist investigated the population growth of bacteria. The culture solution. At the start of the investigation, the culture solution contained all of the nutrients the bacteria needed. The scientists determined the number of living bacterial cells in the solution every hour for over 2 days. Here's the apparatus. You've got your culture bottle. So instead of doing it in a petri dish, it's done in a bottle. And you've got your solution. And you've got your little lid there, your cap. And it's important that they have air holes because, like any organism, the bacteria needs to respire, and it's really important. They have access to oxygen. Okay, describe why they are. Oh, I just answered the question for you guys. To allow air, oxygen in for respiration. No, aerobic respiration doesn't give oxygen. It uses oxygen and produces carbon dioxide and water as a result. Photosynthesis gives oxygen. I don't know whether this is a triple practical or not, but it's coming up in combined, so it clearly must be a thing, unless they made a change since 2019. Okay, give one reason for what is happening to the number of bacteria at the end of the stage. So why does the number of bacteria decline at the end? Or, okay, no, you have to talk about the bacteria at the different stages. So initially, you can see the bacteria increases. So what happens there? Let me go back to this. Okay. So initially, during the culture, the bacteria increases. So at point A, however, there's no change. It's kind of flat. So why is there no change initially? Because there's the reaction has just started, so there's likely to be not much mitosis going on because you've just started the reaction. Then at the point B, what's happening is there's going to be lots more mitosis. Cells are going to be dividing very, very rapidly, and mitosis is going to be happening as the bacteria is spreading and realizing they have lots of nutrients available. And then at point C, we've got this kind of stop in bacteria. That just means that the population is staying the same. The rate of the cells dying equals the rate of cells dividing. And then finally, this kind of going down plateau happens because the cells end up dying. Why did the cells die? Well, different reasons can happen. The nutrients running out, that's probably the main reason why. There's a limited amount of nutrients in the bottle, and therefore they're going to eventually run out of resources. Or you can say there's lots of toxins, there's higher competition, and maybe the bacteria starts undergoing survival of the fittest between each other. The reduced amount of resources available. Okay, that goes through those. Now moving on to this one, which is also talking about surface area to volume ratio. Why is everyone saying 9 a.m.? I'm slightly confused. Maybe talking about the time for your exam. Okay. Continuing on, so we've got surface area to volume ratio. Explain why a multicellular organism cannot absorb all of the nutrients it needs by diffusion through its outer surface. So we already spoke about this. It requires a larger volume to supply to. The diffusion distance is longer in multicellular organisms. And therefore, because the volume required to supply it to is greater, each unit of surface area is greater in the organism, so the diffusion rate per unit of volume is slower in a multicellular organism. Okay, question 5. We've got a root hair cell viewed under a microscope. The root hair cell was viewed at a magnification of 50. The image length of the root hair is 43 millimeters. Calculate the real length of the root hair. So this is literally exactly the same question, apart from just changing the numbers as the other one we did. So you just do, you rearrange the magnification equation. The size of the real object is equal to 43 divided by 50, and that is 0.86, but that's in millimeters. So to convert this to the micrometers, you then multiply it by 1,000 and you get 860. Okay. The next one. A microscope has a 5 eyepiece lens. Describe how to use this microscope to observe a prepared slide of root hair cells at a magnification of times 50. Now, for a detailed view of this, I recommend going to the Tuesday live stream, as we went through all of the possible bullet points and possible errors in this. But just recapping that, you want to place your slide on the stage. You begin by using your lowest power objective lens. You adjust your mirror if it's the old type of light microscope, or you switch your light on so that it passes through the slide. You then move your stage as close to the lens as possible, but you don't want it to touch the lens crack when you're using it. I don't know what is going on in the chat. Okay. You then turn the focusing knob so the slide moves away from the lens. You turn the focusing knob until the image comes into focus, and you then use the fine focusing knob to really improve the resolution and increase the ability to see. Okay, and yes, some people were talking about how you start with the times 10 magnification, and yet you can say that, and that actually does get you a mark as well. Okay, moving on to the next question. Root hair cells absorb water and mineral ions from the soil. A scientist investigated the rates of nitrate ion uptake by 2 seedlings. Okay. The scientists determined the mass of nitrate ions absorbed by each seedling every 30 minutes for 4 hours. And here are the data. Yeah, I don't know what is going on in the chat. Thank you for the people defending. The chat is mental, I agree. They are mad. Okay. That is there. So, describe the changes in the rate of absorption of nitrate ions for the seedling with no oxygen added, using information from table 5. So, changes of the rates. So you can see that initially, let's look at the increases. So it goes up by 45, then goes up by 25, and then goes up by 25. So it's going slowly and slowly. So it's initially got a very fast rate, and then it decreases as it continues. So the rate is fastest in the first 0.5 hours. As you can see, it goes up by 100 initially, which is a lot. And then as the reaction continues, decreases gradually. And then it eventually falls to 0 as there's no change. So that's how you would discuss that. So you would be asked, you would be required to talk about the specific times and how, almost you could draw a sketch of a graph to show how that is changing to get an idea of whether it's increasing or decreasing. Explain what the results in the table 5 show about how nitrate ions are absorbed. So this is a slightly harder question. So we're told, therefore, that well, looking at it, with no oxygen added, it's a lot less, isn't it? Whereas with oxygen added, it's a lot more. So clearly, the use of oxygen or the presence of oxygen increases the amount of nitrate ions that are being absorbed. This then suggests that they are absorbed by active transport because it requires energy, and the respiration uses the respiration requires energy, and therefore made via the oxidation of glucose. And therefore, if it requires lots of oxygen, it's suggesting it's an active process. So why is the presence of oxygen increasing it? So that's a really good question. This is a harder topic, a hard question. So because there's more oxygen, we know that this is it's an active process. It requires active transport. Active transport requires energy. And if we're told that when there's more oxygen, the rate is faster, that then tells us that there's more respiration going on. Respiration going on, then that's suggesting that the more oxygen you have, the greater the active transport is going to happen. I hope that explained it. It's a bit difficult, but hope that says enough. Okay. Nitrate ions are essential for plants to grow. Describe how nitrate ions are used in a plant to help the plant grow. Well, nitrate ions are used with glucose in order to form amino acids. Now, amino acids are important because they synthesize proteins, i.e., they allow growth to occur. Oh, wow. Sorry, my lamp just fell. Okay, I think the chat is really taking the toll of being on this live stream for 3 hours. I'm so sorry. I hope you found this helpful. We are now finished with the live stream. We've done lots of papers, we've done lots of practice. You can come back to it if you want. If you have any more questions, just comment them and I'll reply to them in the future. But I think everybody needs a break after 2 hours and 57 minutes of doing this. I hope you enjoyed the live stream. I think the chat was a bit crazy, but a lot of you were engaging with it in a positive way, answering the questions. So I think that's really good. This is not for foundation. This is for triple and combined higher. This paper was combined higher. We're going through both. So thank you so much, guys, for being. It means a lot. And I will probably see you on the Sunday live stream for, I think it's English literature revision. Okay? See you guys later. If you have any questions, do comment them down below. But I think we all need to have a little bit of a rest. See you later.